There's a lot going on in this lab, and that's true for either doing this in person or doing this online. Um, you're going to learn about the microscope in this lab, which means we're going to have to familiarize ourselves with the parts of the microscope. I've got a video to show you how to use the microscope. I also have a couple of virtual microscopes you can use to kind of play with things. Using the microscope, the parts of using the microscope involve finding the image with the light microscope. And you may want to remember the nice little ditty, find it, frame it, focus it, flip it. So find it, it's you moving it, you're putting the slide in, you're moving the stage up to find to see the image. Then frame it, meaning to put it in the middle. Focus it, get it really into focus because it needs to be in focus. Because when you're going to the next lens, if it's not, the better the image is on the previous magnification, the better it is for the next one. You've got the first, initially you're going to work with scanning power and low power and look at Hydra. Then you're going to look at high power for the paramecium slide. Hopefully by that time you'll have realized a little bit about how these things work, and we're going to look specifically at some of the properties of the compound microscope. We're going to use the letter E activity to learn about image orientation. I'm also going to use the letter E activity to tell you a little bit about field of view. And then we're going to play a little bit with the property of the depth of field, which is usually probably a bad thing, but you can use it to your advantage to try to find which thread is on top. Then we're going to learn about computing magnification with the microscope, and we're going to use the compound microscope to measure a paramecium. Finally, we're going to be looking at microscopic features of leaves, and those are beautiful. And then eventually you're going to watch osmosis happening in Elodea. Osmosis I talk about in the lecture part. So, and I'm also going to share some videos with you for osmosis. But that's the plan for the lab this week. This slide is just to remind you of the parts of the microscope. I go over those in the lecture part in detail, but just to, when we're talking about the things that you need to twirl, this is just a little reminder to help you remember. Also, this or the other microscope may be the images you're going to see in the exam. In your lab handout, you have an explanation of how to put a slide on a stage. That's the first thing you should do. So when you put a slide on a stage, you make sure that the stage is all the way down, and you put the slide in there by moving that lever, by just opening that bracket up and gently putting the slide in there. Students always try to put these slides underneath of the bracket somehow. There's no need for that. You can just put it in there and it'll hold it. It'll hold it tight. The only time I've ever seen a problem is if the slide was wet. So make sure the slides are dry, because if they're wet, they're sometimes sticking to the slide because the slide holder moves, and sometimes if the slide gets stuck at some places due to wetness, it doesn't really work as well. A lot of the information that you're going to be using today or this week for the microscopy lab is going to be on the Google site and in Blackboard. The information is more or less duplicated in both places, so you can find all of it on either the Google site or on the Blackboard site, whichever site you're more comfortable with. One of the pieces of information that's there is there are two different videos for basic microscopy use. The one on the left are scopes that are similar to the ones that we use. It's about 4 minutes long. The one on the right shown on this slide is about 12 minutes long. The scope is different, but the gentleman who does those that explains the microscope basics, he's got a couple of tips and tricks that are kind of cute. But you don't have to watch both of them. Just watch one of them. You know, watch the 4-minute one. That'll get you going. Both the Google site and the Blackboard site also link to two different virtual microscopes. I'm linking to two different ones because one of them actually requires flash, and depending on what kind of computer you're running, you may not be able to use flash. Of course, the flash one is the one that's actually the nicer one. I think here it's the one on the bottom right. That's the one that requires flash. But both of them do more or less the same thing. So you can do some of the activities that I'd like for you to look at using those scopes. When we meet in class, two of the things we look at usually are a slide of Hydra and a slide of Paramecium. Both of those organisms are introduced on the Google site. Hydra is much bigger than Paramecium, and that's usually the one we look at first. And one of the things that a lot of the specimen that we're looking at under the microscope have in common is that they're stained. So when you're trying to focus on any kind of item, you usually are looking for the colored thing, and then you're zooming in from the colored thing. If you're playing around with the virtual microscopes, that's actually something that you can do pretty well. You can just pick any kind of sample and try to find your way to the sample by just starting to look at color and then trying to see how far you can get. One of the first things we want you to understand is that the image that you see under the microscope may or may not look exactly the same way as it looks when you put it there. You won't notice that, right? Because all you see under the microscope, you don't have any reference for what's up, down, forward, backwards. And so one of the first things we have you look at is a letter E. And when you put that down under the microscope, you make a wet mount, and there's instructions how to make a wet mount in both the Google site and the web on the Blackboard site. When you make that letter E, you make the letter E look exactly like it would when you're writing it. And then what I want you to look at is I want you to look at it under the microscope and see what it looks like now. If it looks the same like the original letter, or if it's upside down backwards or mirrored or just upside down. So which one of these four is it under the microscope? And that's one of the questions that you have to answer in your handout. I do talk about why it is the way it is in the lecture part, because there's an explanation for why it is the way it is. The field of view explanation, I think, is done pretty well with the letter E. With the 4x scanning lens, you can see the whole letter E. You see it at the 40x size, 40 times enlarged. With the 10x lens, you see it as a bigger image, but you see less of it, right? And that's because your field of view is your field is limited by the size of your objective, by the size of the openings that you have to work with. And so with each magnification, you see more detail in the image, you see the item bigger, but you see less of the overall image, which makes sense because as you're zooming in, you'll see less and less. Otherwise, you'd have to make a bigger picture. In your questions set, there's a couple of places where you're asked about about total magnification. And that's simply the magnification of the objective lens multiplied by the magnification of the ocular lens. So that means your 4x, your scanning lens, which is 4x, gives you a total magnification of 4 times 10x, which is 40x. And that's, the same thing for the other ones. The next thing we ask you to do is to measure with the microscope. And there are a couple of different places where students usually get hung up when we do this. The first place we get hung up is with the units with the metric units. Let me tell you, let me sing the praises of the metric unit. The metric unit, to get from one unit to the other, you only have to add zeros. You multiply by 10 or by 100. It is so much easier than that stuff you have to work with every day. So let's work with metric here. So for compound light microscopes, the unit of measure that we usually use is a micrometer. Whereas when you see that it says micrometer that U there is actually a mu. But in order to make a mu, you need to enter a symbol that sometimes doesn't get interpreted correctly, and it doesn't get interpreted correctly in this particular program that I'm using. And so when people try to fake a mu for micrometer, they oftentimes put a U instead. So if you see that means micrometer in most cases. 1 millimeter is 1,000 micrometers. So I put a ruler there to try to help you remember what a centimeter is, so that you can have an idea of what a micrometer is in relation to the rest of these measurements. So you see the ruler, and the bottom scale is inches. You see 1, 2, 3 inches on this ruler. On the top, each of the numbers reflects a centimeter. Each of the little divisions between the centimeter, there's 10 of them. Each of the divisions of the little, each of those little divisions is a millimeter. And so then a micrometer is the 1,000th part of a millimeter. So that's tiny, right? That is not something you can see. So the first thing we're going to ask you to do in the lab is to determine the diameter of the field that you can measure on low power. And you do that by literally putting a ruler underneath of there and seeing what you can see. So at low power, you can still measure the field with, like, literally with a ruler. But once you get to the next higher up, you can't measure it anymore because it's so small that you don't see the lines anymore on a ruler. And so we calculate that. And the way you calculate that is you take the diameter of the low power field that you just measured with the ruler, and you multiply it by the magnification of the low power, and then you divide it by the magnification of the high power. And that gives you the size of the field, that you can't measure. So what the diameter of the field is under high power. And that's the first thing we're going to do. And I'm going to walk you through that a little bit because students tend to get really hung up with that one. So the task at hand for this particular activity is to measure the size of a paramecium. So that's what you want to find out at the end, right? So you're trying to figure out what the size of a paramecium is. You're looking at the paramecium in high power. That's the picture on the right. And you're going to figure out how big the paramecium is by figuring out first what the diameter of the field of view is on low power, and then doing the calculation. So when you're putting the ruler, the centimeter side on the ruler, under the low power objective, you can see one line, and you can imagine that the next line is directly past what you can see. So the diameter of this field is therefore, it's 2 of the little lines between the centimeters. Since one of those little lines is 1 millimeter, the diameter of the low power field is therefore 2 millimeters. But we're working with micrometers, right? So the first thing we're going to have to figure out is how many micrometer is 2 millimeters. Well, I told you earlier that 1 micrometer is the 1,000th of a millimeter. So there's 1,000 micrometers in 1 millimeter. So how many micrometer is therefore in 2 millimeters. So that's the first thing you're going to have to figure out. And then we're going to continue this experience to finally figure out what the size of the paramecium is through the high power lens in micrometer. So we just measured the field under low power, which was 10x, right? And we came up with the fact that that's 2 millimeters, and you should be able to figure out what that is in micrometers. Then based on that, we can calculate the field of view of the high power field by taking the number of micrometers from above and multiplying it with 100, which is what the lower power total magnification was, and dividing it by 400. And just because math is scary, let's look at this one more time. So you're coming up with the field of view in millimeters based on what you did with the ruler. You turn that into micrometers by doing the conversion. Then that number in micrometer goes down to the equation where it's multiplied with the magnification at low power and divided by the magnification in high power. And then the result of that, which gives you the diameter of the field at low power, allows you to estimate the size of the paramecium, which is shown in the picture in the middle. When we're in class, we also use a Zebrina plant leaf. On the underside of it, on the underside of leaves, plants have stomata, which are what they use to breathe and to inhale CO2 and exhale oxygen. Interestingly enough, that's on the underside, and it's an opening that opens and closes, and it's guard cells on the wall. They control the opening and the closing. And the Zebrina plants have a beautiful, very, very striking stomata. So, or very, very, that's the plural, very striking stomata. So what I want you to do here is I want you to look at the underside of a Zebrina plant. I want you to count the stomata, which is the green dots. And when you zoom in far enough, you can actually see them, but this happens to be you're visualizing these at low power, so at 10x. And what we want you to do is count the number of stomata in the field of view at 10x. And then I want you to calculate how many you would find on a leaf of the size of a quarter. And so the first thing you have to do that is calculate the area of in the field of a field of view. And just as a little hint, if that number gets very complicated, you're doing it wrong. Because all the numbers should get very, very easy, at least at that beginning part. And then you can measure the area of the quarter. You've got a circle of the correct size in your handout. If you're not printing out your handout, you can actually look up the size of a quarter. Make sure that you pay attention to the radius versus diameter. Those are not the same numbers. So you need to actually do the calculation with the correct one. And then you set up the equation on the bottom to set up a ratio of knowns to solve for the unknowns. So the stomata in the field of view over the area of the field of view, which you can calculate, means that you have x stomata in the area of a quarter. And a lot of these questions you can actually answer in the form of a quiz, which I'm going to show you in just a bit. Last but not least, we're going to talk about osmosis, and you're going to watch the impact of salt on the cells of Elodea. I talk in the lecture a little bit about osmosis, but basically what osmosis is the movement of is diffusion through a semi-permeable membrane, which leads to a net movement of water from a higher concentration of water to a lower concentration of water. And that usually means that water moves into the direction of where the solutes are higher. Osmotic balance means when there's as much water going in as there's out. Um, so that's usually a balance. And in the case of animal cells don't like it at all when they go into hypotonic solutions because they will fill up and they burst. Plant cells are the opposite. Plant cells have cell walls, and because of that, they actually like hypertonic water. They really don't like hypertonic water, so they really don't like salt water. And we're going to look at what happens when you put Elodia into salt water. And if you're not going to be able to see this in class, there's a video of it for you on the Google site. What we do in class is we prepare a wet mount of an Elodia leaf using tap water. We look at one of the cells, and you can zoom up and down, you can scan up and down to kind of get it into focus, but usually you'll see a couple of different layers of plant cells. And then there's a second wet mount that we prepare using 6% salt water, and then focus on those and see what happens. And what happens, and you can see this in the difference between the two pictures on the left and on the right. The one on the right is the one with the salt water. And what you can see is on the left, you see the little cells, which are like the little squares, right? And all plants have vacuoles that basically fill out the entire cell. And the chloroplasts, they look like they're just running around the edges because they're running around on the outside of the vacuole. Whereas if you put a plant in salt water, the cell shrinks, and what shrinks is actually the vacuole. And so what it looks like, what it ends up looking like, is like the chloroplasts are kind of bunched together in the middle, and that's what you kind of see on the right hand side. And that was it for this week's class.